Podcast is now starting. All attendees are in listen-only mode.
Thank you for joining us today for our webinar on novel technologies for understanding metabolomic communications between host and microbiota. Our speaker is Dr. Alex Bucco, Vice President of Clinical and Preclinical Applications. If you have questions, please use the question box to type them in, and we will do our best to address them either there through the chat or at the end of the talk. I'll now hand it over to Alex, and we can get started.
Okay, thank you, Laura. Welcome, everyone, and thank you for joining us today. I'd just like to start out with a brief bio of your speaker today. I did start out my career at CBER back in the early 80s, which is the Bureau of Biologics and Biophysics. I spent 18 years at Abbott Labs as head of structural chemistry. Then went on to Biogen for another 10 years as Senior Director of Translational Medicine. And I've been with HMT now for about 6 years, working with preclinical and clinical studies with pharmaceutical companies, biotechs, and medical centers. Today's talk will focus on the microbiome and our ability to use CEMS to measure metabolites in various sample types. And I want to say that the microbiome is something that's not just a business proposition, but something that all of us may have an interest in, as the association between the microbiome and our health starts to gear up, and we learn more and more about that association. The agenda for today will be talk a little bit about our strategy for how to approach measuring the microbiome, the type of specimens that we can measure, and define the challenges, which is about the diversity of chemical species and the advantages of the CEMS to look at various classes of these metabolites in terms of how we measure them and quantitate them, and how we can associate some of these metabolites to clinical phenotypes. And then I'll end with a short talk about the company itself, Human Metabolite Technologies. Our strategies for the microbiome is really to integrate the CEMS data with bacterial sequencing data and clinical data, to really merge all that data in order to best understand what type of reported metabolites are associated with clinical phenotypes. And then look at these perturbations of these metabolites due to either therapeutic intervention or diet or disease. We also want to work with various things, including fermentation products, animal models, or clinical samples, in order to achieve these goals. Our targeted areas include, again, fermentation products, skin and skin tissues, hair follicles, sweat, a lot of each of those biofluids, tears, serum, and plasma, as well as look at fecal matter, both solid extraction and liquid extraction, well, depending on the type of microbiome that might be of interest. What are these metabolites? Again, these are generally considered as nutrient supplies. It's interaction between the bacteria and the host. It could be diet generated, or it could be from epithelial tissue, adipose tissue. And what the bacteria does is transforms these organic molecules, proteins and lipids, and fats, into carbon and nitrogen, energy and biomass for the bacteria. And of course, also produce waste products or uremic toxins. These sea of metabolites then can interact with the host in terms of signaling, and they can interact with the lymphoid system, our nervous system, inflammatory system. Some of these metabolites are bacterial or fungal specific, but many are endogenous but affected by the bacteria themselves. These specific small molecules or metabolites associated in the microbiome run a large range of chemical families. They include amines and polyamines, organic acids. Many of these are polar and charged, phosphates, sulfates, small chain fatty acids, osmolytes, which come in many different varieties, amino acid and their derivatives, as well as longer chain unsaturated and fatty acids. Again, these are small polar charged molecules to more hydrophilic or neutral molecules. And then what's important too, that these particular molecules can interact with a variety of different pathways in terms of energy metabolism, glucose metabolism, oxidative stress, purine metabolism, amino acid metabolism. The challenges then in measuring these metabolites is that they do run from very small under mass of 100 up to very large, up to 1,000. Many of them are isobaric, meaning they have the same mass and maybe chemical formula, but different structure. They can have branch versus linear, for example. They often are very highly charged, sulfates, phosphates, guanidino compounds. All these in one complex mixture that has to be separated. So you can imagine that there's not one technology that will do it all, so multiple technologies would be required. Our strategy then is to apply capillary electrophoresis to separate these mixtures in combination with high mass accuracy mass spectrometry to provide, and also to provide quantitative solutions. And then take these profiles and link them to a clinical phenotype in order to develop biomarkers for disease or in drug development or treatment of disease. To implement this strategy, we provide quantitative measurements then along with relative measurements. We also provide spatial resolution by providing a isocratic capillary electrophoresis. And high density metabolites is provided by the separation using a large growing small molecule library that we've invested into the HMT. Again, using capillary electrophoresis, we're able to, in exact mass, we are also able to provide novel discovery, looking at identifying metabolites that have not been observed before. We also collaborate with our clients in terms of working with them on pre-planning any experiment, as well as delivering a statistical and biological interpretation at the end. And we do have a large library of metabolites, many of them validated, some listed as unknown, and some predicted. And we use these in order to annotate our measurement of our metabolites. Our migration time is accurate, and we use this to help predict unknown metabolites. And our resolution from the CE allows us to separate metabolites that have the same mass. On the far right, you see one metabolite series of 132, 1017, isoleucine, leucine, alloleucine, and orleucine. Again, these are all isobaric, they're just different structures, and we can separate them by the retention times, as you can see underneath. Below is another series of 3 metabolites of the same mass, again separated by migration time using the high resolution capability of the capillary electrophoresis. And this migration time is predictable based on the properties, physical properties of each of the metabolites. So our library is broken down into families in terms of nucleic acids, amino acids, fatty acids, short-term fatty acids, and then from that superfamily we can predict a migration time based on any changes to that structure, oxidation, methylation. And again, as I mentioned, we provide quantitation, and we can quantitate up to 400 metabolites at the same time, provide on-targeted and global profiling. These are some examples of some metabolites that are isobaric. You might be familiar with isobutyrate and butyrate, for example, alanine, sarcosine. Also exemplifies that the CE is very good at looking at small molecular weights, urea, ethanol, and gloxylate as well. We also can identify various gamma glued dipeptides and separate those from the dipeptides themselves due to the resolution capability of the capillary electrophoresis. And we can separate and identify all the glycolytic intermediates using capillary electrophoresis as well. The ones highlighted in blue are on the next slide. In this paper, we were looking at senescence in mice. We were actually started out looking in human samples, human feces, and looking for metabolites that were had concordance with putrescine. And we've identified several metabolites, including arginine. And arginine supplementation into mice did show an increase in lifespan and an increase in learning capability by supplementing the diet with arginine. But again, in the study itself, we see several metabolites identified, 4 of them which are actually isomers. So just showing the point that we need to develop high resolution techniques to make sure we can specifically identify the metabolites that we see. Yeah, in our small library that contains knowns, unknowns, and predicted, contains many different types of molecules, from coenzyme acylcarnitines, short-chain fatty acids, middle-chain fatty acids, phosphates, nucleic acids, amines, midazoles, organic acids, and a large number of peptides that include gamma-glupeptides, cysteine adducts, glutathione adducts, lactosyl adducts, fucosyl adducts. as well as novel discoveries to give you a highly annotated metabolome profile. Reported metabolites are metabolites that we've associated with different clinical phenotypes. So with odor, for example, polyamines are generally what's associated with odor. So these are basically from amino acid degradation, decomposition. They're responsible for odor and malodor, but they're also indicated an inflammatory response and skin sensitivity, and they also are affected by age. Lactic acid is also associated with certain bacteria, and this comes from the glycolysis and glucose metabolism pathway. It could also be microbiocidal, so it can affect the wellness of a bacteria in the microenvironment. Lactic acid also contributes to acidosis in the cytosol and also can induce osmotic stress. It also be related to immune reaction with the host. Immune response to indoles and taurine often are associated with it. These are a series of indoles and amines and unsaturated amines, kynurenine pathway. Again, these AA arch indole ligands, again, are responsible for working associated with lymphoid cells and epithelial cells, modulating the immune system. And along with polyamines, these also decrease with age. Taurine itself is also associated with inflammation. Osmotic stress is an important clinical phenotype that we mentioned associated with charged metabolites such as betanes, family, amino acids, and carnitines, as well as amines and precursors, all associated with the stress response. This is usually associated with cytoprotective response and osmolality of the extracellular medium. Pain and itching also again associated with polyamines and both charged polyamines and charged acids, so as well as polyols, urea, uric acid, amines, diethylene glycol, and serotonin. Oxidative stress, another large family, a host of molecules associated with that. The short-chain fatty acids are often discussed with the microbiome, and these are usually carbon 2, 3, and 4, and 5 carbon length sugars. Oxidized polyols, sarbose, and oxidized products, and glycosides as well. And these are usually connected to immune system through T cells, associated with energy in epithelial cells, affecting autophagy, also linked as well to inflammation. And looking at the short-chain fatty acid story, again started out very many years ago, associated with irritable bowel syndrome. Today, again, refers to other interactions with the host, including immune functions. We know the transporters are MCT1 and SMCT1 associated with that, and as well as the GPCR receptors. It's interesting to know, if you look at the publication history of short-chain fatty acids, it's growing since the early 60s in association with the interests associated with the microbiome. Though you can see here in the early 70s, there's a large bump, which is kind of unusual when you look at these types of publication history from PubMed. And we found out this is actually associated with thin layer chromatography, if you search that. And during this time, the commercialization of these plates became very popular, and it became a popular way to use the silicate to separate out these fatty acids and use chemical ionization mass spec to really identify them. So there's a large boost here in these publications. However, the association with specific bacteria was lagging until 16RS came along. And then the ability to associate better these short chain and middle chain fatty acids with specific bacteria became prominent. So back then in the 70s, though, again, FTM&R, chemical ionization mass spec, back to some earlier bacterial sequencing and thin layer chromatography were the enabling technologies in order to spur this research on. Today, the microbiome work is skyrocketing due to the advent of, again, RNA sequencing, public sequencing databases, as well as metabolite databases. So these, in conjunction with CMS, now are becoming enabling technologies to be able to measure and associate a specific microbiome with the classification provided by the RNA sequencing. So I just wanted to mention at the end that with Metabolomics, the metabolomics that we've been providing from HMT has started in 2003. We started going global in 2013 with offices in Boston. We expanded into Europe and just last year with an office in Leiden. And we also have an office in Yokohama for a biomedical development group. We have, aside from our global offices, we have distributors in Korea, in China, and Taiwan. Our capillary electrophoresis systems, again, are focused on using open capillaries. And so this makes the exchange of capillaries very easily done. We don't have any issue with standardization. These are just few silica columns, 30,000 volts goes across. And the important point is that these metabolites are separated by charge and physical properties. So it allows us to have clean separations, high resolution separations. And we have a large laboratory in Japan that conducts these analyses. Knowing that we have both, we can cover very well this charged metabolome space with CEMS. We also have LCM options, so we can cover both lipophilic and space as well. And to do that, we have options including MScan, which is looking at amino reactive and lipids. We have 400 target lipids. We offer a dual scan, which is an LCM process. So this allows us to look at the larger chain lipids, steroids, and bile acids. But we also have all the CE methods here to look at the smaller fatty acids. And we have our untargeted profiling options. We have our quantitation options. We also can look at isotopomers using our F-scope for C13 and 15 labeling. So we have many options available here for depending on the types of samples and type of goals within each clinical project. So to summarize, where our goals are to develop tests and to stratify patient subsets by associating various metabolite profiles with clinical phenotype, and look for best ways to predict treatment modality and outcomes based on these associations of metabolomics and phenotype again. So through our business model really includes then providing this high resolution CEMS technology to provide both a quantitative and an untargeted metabolic profile. To be able to use this high separation to look at isomers and charged species and polar species, and to use a large and growing small library to highly annotate these profiles and provide an opportunity for novel discovery. And from this, develop phenotypes that are linking metabolite profiles with bacterial associations, and devise a business model for collaboration. So that ends our short talk today. Just as an introduction, I encourage you to connect with our website in the area from which you're coming from, the USA, Europe, and Asia, and connect with them for any further information.
Thank you, Alex, for that. And we do have a couple of questions here that we would like to address online. So one of the questions is, how does CE compare to helic columns?
The helic column is a reverse phase packed column technique designed for charged species, similar to what the CE can do. However, again, you're still working with a packed column, so the resolution is not as high as you would get with the CE. So our ability to resolve isomers is heightened with using the high resolution capability of the capillary system versus the reverse phase from the helic. And that would be the, and in addition, the CE, aside from a high resolution, is a different technique, so we actually see different types of molecules as well. So in the molecules that overlap, we have better resolution, but also we see new molecules that will not be eluded off or seen with the helic column.
Great, thank you. How long does it take to get results?
Well, we typically tell everyone 4 to 6 weeks. It just depends on the size of the study. Usually the deliverable at that time will be an Excel file with all your data, raw data in it, and a deliverable, which a PDF, which would be a short analysis, complete analysis of your study. Typically, following that, every client has a scientific liaison. So within a week or two after receiving your data, you'll have a personalized a meeting with our scientific liaison who will provide more details and go over the results with you.
Another question we have, are all of our labs for analysis in Japan?
Currently, all of our analysis labs are in Japan. We do offer shipping from our regional offices, so samples in the northern hemisphere get sent to Boston, in Europe go to Leiden, and then we handle all the shipping of the samples directly to our lab. Usually takes 2 to 3 days. Those clients can ship directly to Japan if they want to.
One other question that we can address today, what kinds of specific protocols do you have for isolating bacterial metabolites?
Well, each specimen type has an extraction protocol, so that's serum, other types of biofluids. The only thing that might be unique would be in fecal matter. There we do have choices in whether to extract from the solid material or look at the liquid portion of the fecal matter. So we have protocols that we actually will send to each client based on the type of sample that we have. We also have a customizable approach, and so if we have something that's unique, tears from rats, for example, or we can work with you in terms of trying to optimize that protocol through a pilot study. So though we have protocols for every type of sample, we're willing to work with you on customizing something that might be unique.
How many samples are required for a typical run?
Well, that's a good question, and there's no one answer because it depends on the types of samples and the goals of the study. So usually for cell line work, which may not be here in the microbiome, but since those are fairly reproducible, the numbers are pretty small. 2 or 3 samples per group usually will suffice. For animal studies, again, you can have quite a bit of diversity in your animal model. And so again, the group size might be anywhere from 5 to 6 up to 10 to 12, depending on the diversity within the group. For human samples, again, it gets controversial in terms of what's the minimum size. But I tend to think that anywhere between 25 and 50 per group is what's necessary for a study that's going to get you some results that are statistically significant. Just because of the biodiversity of the human species and the diversity that we're going to see. You can certainly work with smaller groups for pilot studies to try to get information about differences and depth of analysis. So this is something that we also would work with each client in terms of developing the correct work plan that would have sufficient impact. So this is really done by case-to-case basis. But we have no minimum samples, but small sample sizes are generally just used for feasibility studies and small pilot studies.
Well, that is about all of the time that we have today for questions. We really appreciate everyone for attending. And if you have any additional questions, feel free to reach out to your local office. And you can email us or contact us with the numbers on the slide that's still up. And you will also receive copies of this webinar from Microbiome Times. Again, thank you very much for attending.